Today I am going to explain about topic of pteridophytes. So pteridophytes, the meaning of term pteridophytes is pteron. Pteron means feather-like appearance of plants. The plant body or leaves of the pteridophytes appears feather-like appearance. So cherries. Cherries means fern plants. The higher pteridophytes is nothing but fern plants. So pteridophytes, the leaves, the plant body appears like a feather-like condition. That's why this is called pteridophytes. The term pteridophytes was first given by Ernest Haeckel, scientist. The term pteridophytes was given by Ernest Haeckel, scientist. The study about pteridophytic plants is known as a pteridology. The study about pteridophytic plants is known as a pteridology. Pteridophytes are first land terrestrial vesicular plants. So first land vesicular plants. First land vesicular plants are pteridophytes. That means vesicular tissues, xylem and phloem originated, evolved in pteridophytic plants. So conductive tissues are present in pteridophytic plants. But these are cryptogamic plants. Cryptogams. That means non-flowering plants. So flowers and seeds are absent. So tracheophytic plants, vesicular plants. First land terrestrial tracheophytic plants are pteridophytes, vesicular cryptogams without flowers and seeds. But these plants would not achieve desired success due to some reasons. So what are that reasons? So pteridophytic plants are first land vesicular plants, but these plants did not spread, could not attain or achieve desired success on land. So why? Because some reasons are there. So first one, spores. So generally, sexual spores are producing in pteridophytes specialized structure, that means sporangium. But for germination of these spores requires a wet condition. Why? Because pteridophytic plants generally growing found at cool, damp, shady localities and wet condition. So for germination of spores also requires the wet condition. These spores cannot germinate in dry habitats in dry condition. These are requires the wet condition. Next one, by the germination of these spores, haploid, these spores are haploid condition. Haploid spores germinate, then produce the haploid gametophytic body. Haploid gametophytic body, nothing but prothallus. Prothallus. These prothallus also requires cool, damp and shady localities for growth into adult plants. So these prothallus is a susceptibility to desiccation. Susceptibility to desiccation. Why? Because this prothallus requires the cool, moist condition for growth and development. It is a susceptibility to desiccation. Due to these requirements, these pteridophytes not spread in all areas, so limited spread in a condition. These are growing narrow geographical areas only. At the same time, for fertilization, sperms anthrozoids move and reach archegonium. That means fuse with the egg cell. So these requires a thin film of water. That means the movement of the sperms requires to a thin film of water. Due to these reasons, this pteridophyte could not achieve the desired success. Next one, general characters. So general characteristic of pteridophytes. So pteridophytes generally growing at moist, wet, shady localities and flourish sandy soil condition. So pteridophytes generally growing at wet, moist, shady localities, then flourish and sandy soil condition. So pteridophytes generally growing moist, wet, shady localities, flourish and shady localities. The plant body of the pteridophytes differentiated into true root, stem and leaves. True root, stem and leaves. So root is a adventitious roots are present. So root system is a adventitious root system. Then stem differentiated into epidermis, cortex and stem. Epidermis, cortex and stem. So due to vascular bundles are clearly differentiated, originated in pteridophytic plants. That's why stem is differentiated into epidermis, cortex and stele. So stellar evolution originated in pteridophytic plants. So stele is a center core of the axis. This stele includes vascular tissues, xylem and phloem. Then in pteridophytic plants, in all plants, 4 types of steles are present. So these are protostele, siphonosphere, then solenosphere, then dictyostele. So 4 types of steles are present. So protostele, siphonosphere, solenosphere and dictyostele. So first one coming to protostele. So protostele is a primitive type. So it all present in all types of the plants. In protostele, xylem located as a center core. It is surrounded by phloem, then pericycle, then endodermis, cortex and epidermis. So protostele further 4 types are there. So first one, haplostele, actinostele, third one plectostele, then mixed protostele. So protostele further 4 types are there: haplostele, actinostele, plectostele and mixed protostele. So first one coming to actinostele. So in actinostele, xylem is occupies like a central cylinder-like condition. It is surrounded by phloem, then pericycle, then endodermis, then cortex is present. So it is a xylem, phloem, then pericycle, then endodermis, cortex, and epidermis. So pith is absent. That means medulla is absent. So in haplostate, xylem located as a center core. Medulla is absent. Then xylem surrounded by the phloem. Then pericycle. Then endodermis. Then cortex. Then epidermis. This known as a haplostele. Best examples. Lycopodium, then Xylotum. Lycopodium and Xylotum example for this haplostele. Then actinostele. In actinostele, xylem present as a star-shaped condition. Xylem present as a star-shaped condition. It is surrounded by phloem. In actinostele, xylem present as a star-shaped condition with radiating arms. It is surrounded by the phloem, then pericycle, endodermis, cortex, and epidermis is present. This is known as a actinostele. So lycopodium, some species of lycopodium. In some species of lycopodium, actinostele is present. Then plectostele. In plectostele, the xylem broken into a parallel strips like a condition, a parallel plate like condition. These parallel plate are immersed in a phloem. So in plectostele, the xylem broken into a discrete, a plate-like condition. These plates are immersed in a phloem. This is known as a plectostele. In plectostele, xylem broken into a parallel plates-like condition. These plates are immersed in a phloem. It is a phloem. These are broken xylem plates. These xylem plates are immersed in a phloem. This type of stele is known as a plectostele. Some examples, some species of lycopodium. Next one, mixed protostele. In mixed protostele also, the xylem embedded as a discrete plates like condition in mass of phloem. This is known as a mixed protostele. Some species of lycopodium. Next one, coming to siphonostele. So protostele with a medulla. So generally in protostele with medulla is absent. So protostele with the medulla, this is known as a siphonostele. In siphonostele, medulla pith located as a central core like a condition. It is surrounded by xylem, then phloem, then pericycle, then endodermis, cortex, and epidermis. This is known as a siphonostele. So in siphonostele, medulla found as a central core. Then it is surrounded by xylem. Then phloem is present, then pericycle, then endodermis, then cortex is present, then epidermis. So it is medulla. That means put. Next one, xylem. It is a phloem, then pericycle, then endodermis, cortex, then epidermis. Cyclostele means protostele with a medulla. That means pith. So pith located as a center core. It is surrounded by the xylem. Then phloem, then pericycle, then endodermis, then cortex, then epidermis is present. Leafy gaps are absent. This is known as a siphonostele. This depending on arrangement of xylem and phloem. This siphonostele is two types. So first one is ectoploic siphonostele, then amphiphloic siphonostele. Ectoploic siphonostele, then amphiphloic siphonostele. In ectoploic siphonostele, phloem located external to the xylem only. That means only one side is present. Then in amphiphloic siphonostele, phloem located both sides of the xylem. That means external and internal condition is known as a amphiphloic siphonostele. Next one is a solenostele. So siphonostele with the leafy gaps. So generally leafy gaps are absent in a siphonostele. So siphonostele with the leafy gaps, this is known as a solenostele. So leafy gaps are present. So this type of leafy gaps are absent. These gaps are absent in a siphonostele. So it is a phloem. Then xylem, pericycle, endodermis and cortex is present. This is called the solenostele. Solenostele further two types, ectoploic solenostele and amphiphloic solenostele. So here phloem is present. So external to the xylem, then internal to the xylem. Both sides are present. If phloem present only external to the xylem, this is known as a ectoploic solenostele. If phloem present both sides external and internal to the xylem, inner side of the xylem, then outside of the xylem, this is known as a amphiphloic siphonostele. So lycopodium, some species of lycopodium examples for solenostele. Next one, dictyostele. In this leafy gaps are scattered condition. That means limited number of few number of leafy gaps are present. Next one, in dictyostele more number of leafy gaps are present. Due to more number of leafy gaps, so broken the vesicular bundles, break the vesicular bundles in small strips like condition, small pieces like a condition. These pieces is known as a meristeles. This is called dictyostele. In dictyostele number of leafy gaps are present. These leafy gaps breaks the vesicular bundles into smaller plates like condition, pieces like condition. These pieces is known as a meristele. So these are the leafy gaps. Due to more number of leafy gaps, vesicular bundles are broken into small plates. These plates is known as a meristeles. This type of vascular bundle is known as a dictyostele. So number of leafy caps are present. They are broken the vascular bundles into smaller plates, smaller parts. So this is known as a meristeles. This dictyostele is two types. So first one eustele and attactostele. Eustele and attactostele. So in eustele, vascular bundles are arranged parallel to each other. So vascular bundles are arranged parallel to each other. These vascular bundles are separated by medullary rays. This type of vascular stele is known as a U-stele. This majorly present in dicots and gymnosperms. So in U-stele, vascular bundles are arranged parallel to each other. These vascular bundles are separated by medullary rays. Examples gymnosperms and dicots. Next one, attactostele. In attactostele, vascular bundles are scattered in a manner, not in a particular order. So completely scattered in a manner. This is known as a attactostele. Examples monocot. Next one, leaf. In pteridophytes, leaves are simple leaves or pinnately compound leaves are present. Simple leaves or pinnately compound leaves are present in pteridophytic plants. Generally, in bryophytes, the plant body is gametophyte. It is a dominant phase in bryophytic plants. But in pteridophytic plant, plant body is sporophyte. Plant body is sporophyte. It is a dominant in a pteridophytic plant body in pteridophytic life cycle. Sporophyte is two types. So it is a megaphilous and microphilous. Megaphilous and microphilous. In pteridophytes, sporophyte is two types, megaphilous and microphilous. So in megaphilous, leaves are very large in a condition. Best example is fern plants. In fern plants, leaves are large in condition, megaphilous. Then microphilous, leaves are small. First example, Selaginella. In Selaginella, leaves are small. That means leaves are two types, microphilous, small leaves in Selaginella, then macrophilous, megaphilous in terrace and ferns. Leaves are large. So leaves are simple leaves or pinnately compound leaves, small leaves and large leaves. Large leaves found in terrace fern plants, then small leaves in Selaginella. Next one, secondary growth is absent in pteridophytes. Secondary growth absent. Secondary growth absent in pteridophytes. Then pteridophytic plants are reproduced by vegetative reproduction, then sexual reproduction. Vegetative and sexual reproduction. Vegetative reproduction by fragmentation, then adventitious bud formation. Adventitious bud or branches formation, then bulbils. Fragmentation, adventitious bud, branches formation or bulbular formation. Next one, asexual reproduction. Vegetative is nothing but asexual. Pteridophytic plants reproduce by vegetative, asexual and sexual. Asexual reproduction mainly by the spores. Asexual reproduction by the spores. These spores are produced in a specialized structure, sporangium. Spores are producing in specialized structure, sporangium. That means sporangia. These sporangias are found in leaves, maybe lower surface of leaves. Sporangias are subtended in leaves, on lower surface of leaves. These leaves is known as a sporophylls. This leaves is known as a sporophylls. So asexual reproduction by the spores. These spores are producing in structure sporangium or sporangia. These sporangias are found on lower surface of leaves. So in pteridophytic leaves, due to this reason, is known as a sporophylls. These sporangia grouping to form a one structure. This structure is known as a sore or a sorus. Each sorus may contains 5 to 6 sporangia or 2 to 6 sporangia are present. Sore. Then in sporangia 12 to 16 spore mother cells are present. In each sporangia 12 to 16 spore mother cells are present. Union condition, that means a diploid condition. These sporangia covered by a single layer of a jacket. Single layer of a jacket is present. Marginal cells of the jacket is known as a annulus. Marginal cells of jacket is known as a annulus. These annulus will be useful to dispersal of spores. Then the rest of the cells is known as a stomium. Rest of the cells is known as a stomium. Then these are spore mother cells. So spore mother cells undergoes to meiotic division, then produce the haploid spores. Spore mother cells undergoes to meiotic division, then produce the haploid spores. Then these spores germinate to produce the gametophytic plant body. Gametophytic, pteridophytic plant body, asexual reproduction. Upper epidermis, lower epidermis, then mesophyll. Then sporangia. Grouping of sporangia is known as a sore. Then these sore covered by one wall layers, protective layer. This is known as a indusium. True indusium. Indusium is nothing but a protective coat of a sori. Is known as a indusium. If the sorus covered by one membranous protective coat, is known as a true indusium. In some plants, the leaf margins are reflected and provide the protection to this sori. This is known as a false indusium. True indusium and a false indusium. True indusium means the sori is surrounded by, protected by one protective membranous covering. There the leaf inverted, that means reflected margin and provide the protection to sori is known as a false indusium. There spores generally in bryophytic plants, all spores are similar condition, that means a homosporous condition, but in pteridophytes both a homosporous condition and heterosporous condition is present. That means production two kinds of spores. In some pteridophytic species homosporous condition, in some pteridophytic species heterosporous condition. Heterospory. So heterospory means production or formation two kinds of spores. This is known as a heterospory. Heterospory was first originated in pteridophytic plants. Up to bryophytes, only all spores are similar condition. That means a homosporous condition. So here two types of spores. That means microspores and megaspores. Microspores and megaspore. So microspore is nothing but male spore. Microspore means male spore. Megaspore means female spore. Small and large. Megaspore means a female spore, then microspore means a male spore. This microspore producing in a microsporangium. Megaspores are producing in a megasporangium. Two types of sporangium, microsporangium and megasporangium. By the germination of these microspores due to male gametophyte. Then by the germination of these megaspores will give to female gametophytes. Two types of gametophyte, male gametophyte and female gametophyte. Microspores male spores and megaspores female spores. This is known as a heterospory. Heterospory originated in pteridophytic plants. These lead to seed habit. This lead to seed habit. Seed habit originated in pteridophytic plants without seeds, without the formation of seeds. Some advances towards the seed habit in pteridophytic plants. Best example, Selaginella. So Selaginella shows some advances towards the seed habit. So what are that advantages? So first one, generally these spores are producing in a sporangia. Two types of spores. Two types of spores, microspores and megaspores. By the germination of these two types of spores produce two types of gametophytes. Two gametes. So advances towards the seed habit in Selaginella. What are the advances? So two types of spores, that means microspores and megaspores. Then by the germination of these spores develops two types of gametophyte, male gametophyte and female gametophyte. In pteridophytic plants, two gametophytes are independent in nature. Only in pteridophytes, two gametophytes are independent in nature. Then development of this megaspore within megasporangium. Development of megaspore within megasporangium. Then development of structures like a pollen chamber, stigma to accept the pollen grains. So these initiated this type of structures in pteridophytic plants. So development retention of structures, pollen chamber, stigma to accept the pollen grains. Then development and retention of embryo, so within a megasporangium. So these are the advances towards seed habit. Heterospory in pteridophytic plants generally observed in Selaginella. Then Salvinia, Azolla, then Marsilia, Isoetus. So heterosporous conditions: Selaginella, Salvinia, Azolla, Marsilia, and Isoetus. Then homosporous condition: Adiantum, Lycopodium, Equisitum, Xylotum, Pteridium, etc. By the germination of these spores, microspores and megaspores will develop into gametophytic pteridophytic plant body. This gametophytic is known as a prothallus. Gametophyte is known as a prothallus, meaning higher pteridophytes. That means fern plants. So prothallus is a gametophyte. It is a multicellular condition, green, photosynthetic nature, independent autotrophic condition. But this requires the cool, damp, shady localities for growth and development. That's why pteridophytes spread in limited areas only, growing in narrow geographical areas. These gametophytes are independent in nature. So generally, this gametophyte appears heart-shaped condition. Gametophyte appears heart-shaped condition with one apical notch. It is a multicellular green in condition, parenchymatous condition. So parenchymatous body is parenchymatous body, rhizoids. So unicellular rhizoids are present. So then below this apical notch, cushion like parenchymatous body is present. On this cushion, female sex organ archegonium is present. Archegonium. Then antheridiums are present at the rhizoidal place or between the rhizoids. Antheridium. So antheridium, archegonium both are present on a prothallus. So prothallus is a monoecious condition. Prothallus is a monoecious. So prothallus is a haploid condition, gametophyte. So it is a multicellular, green, photosynthetic, autotrophic nature, independent condition. So it appears heart-shaped with apical notch. Parenchymatous body is present. Both male and female sex organs, antheridium, archegonium present on this prothallus. So archegonium present in upper side of the prothallus. So below this apical notch and cushion like body, nothing but a placenta. Next one, antheridiums are present between these rhizoids. Rhizoids are unicellular condition. These rhizoids will be useful for absorption of water and minerals for perform the photosynthesis. Then sexual reproduction of pteridophytes by producing sex organs. That means oogamous type. Oogamous type of sexual reproduction by the development of sex organs, male sex organ antheridium, then female sex organ archegonium. Then first one coming to antheridium. So antheridium generally hemispermic or globose shape. Hemispermic or globose shape in a condition. Generally, in bryophytes, antheridium is a stalked, jacketed, multicellular condition. But in pteridophytes, antheridium is a stalkless condition, but a jacketed condition, multicellular condition. 3 cell of jacket is present. This also sterile cells. In pteridophytes, antheridium is a stalkless, globose, hemispermic condition. 3-celled jacket is present. In this 32 to 48 sperm mother cells are present. In antheridium, 32 to 48 sperm cells are present. Then these sperm cells are produced. Sperms are antherozoids. Sperms are antherozoids. These sperms are antherozoids are multiciliated or multiflagellated condition. So after mature, these antherozoids are sperms released from antheridium, then reach to archegonium by the help of water. Then archegonium. So archegonium generally flask-shaped condition. That means characteristic features of archegonium we have discussed in bryophytes, similar to that only. So flask-shaped condition. This archegonium embedded in upper side of prothallus. So this in flask-shaped archegonium stalk, venter and neck is present. Neck, then venter is present. In neck, 3 rows or 5 rows of wall layers are present. In neck, 3 to 5 rows of wall layers are present. Only one dioecious, only one binucleate. Only one binucleate neck canal cell is present. So within neck only one neck canal cell is present. It is a binucleated condition. Then in venter, one venter canal cell, then one egg cell is present. So archegonium is a flask shaped condition. It is a differentiated to maybe similar to stalk, venter and neck. So neck surrounded by 3 to 5 rows of wall layer cells. Only one neck canal cell is present. It is a binucleated condition. Then in venter, one venter canal cell, then one egg cell is present. After mature, this egg cell, this venter canal cell and neck canal cell disintegration, degeneration takes place, then release the mucilage. So while this time the matured sperms will release from antheridium and swim in water, reach to mouth of archegonium. So mouth of archegonium has mucilage chemical. So these sperms are attracted by this mucilage. This mucilage may contain the malic acid. These mucilage may contain the malic acid. So sperms are attracted by this malic acid. Then enters into neck, then reach to venter, then fuse with the egg cell. So fertilization takes place. Results of this fertilization then produce the zygote. So zygote is a diploid condition. Then zygote immediately undergoes to mitotic division. Then develops into embryo. Then embryo develops into sporophyte. Embryo develops into sporophyte. These sporophytes depends on gametophyte at the initial days only. Then it is independent in nature. Then sporophyte develops into adult pteridophytic plant body. So these pteridophytes shows the heteromorphic heterologous alternations of generation. Heteromorphic heterologous alternations of generation. That means sporophytic generation, then gametophytic generation alternate to each other. Both are morphologically distinct in a condition, alternations of generation, but sporophytic generation is a dominant condition. This gametophyte may be present on a sporophyte. These pteridophytic plants shows the apospory. Then apogamy. Pteridophytic plants shows the apospory and apogamy. Apospory means a sporophyte directly develops into gametophyte. Sporophyte directly develops into gametophyte. So gametophyte is a 2n condition. That means diploid in a condition. Apospory means sporophyte, diploid sporophyte directly develops into gametophyte without any spore formation. Without spore formation, diploid sporophyte directly develop into diploid gametophyte is called apospory. Then apogamy means gametophyte directly develops into sporophyte. Generally, gametophyte is a haploid condition. Haploid gametophyte directly develops without production of gametes, without fertilization, directly develops into sporophyte. Directly develops into sporophyte is known as a apogamy. Here sporophyte is a haploid in a condition. In pteridophytic plants, both the gametophyte and sporophyte are independent nature. Heterospory first initiated in pteridophytic plants. So heterospory means two types of spores, microspores and megaspores. In homosporous only one type of spores. In heterospory two types of gametophytes, male gametophyte and female gametophyte. So in homosporous only one type of sporangia. Here two types of sporangias, microsporangia and megasporangias. In homosporous there is no importance of evolution. In heterospory there is an importance of evolution. These lead to seed habit. Generally seed habit is a prerequisite. So pollen chamber, stigma, etc. So pollen chamber, stigma may be initiated in pteridophytic plants, but without a seed formation. Seed habit is absent in pteridophytic plants. Then life cycle of based on characteristic features, these pteridophytic plants are classified into four classes. So first one, Xylopsida, that means xylophyte. Lycopsida, lycophyte. Sphenopsida, sphenophyte. Then Pteropsida, Pteridophyte. So based on characteristic future, pteridophytic plants are classified into four classes: Xylopsida, Xylophyte; Lycopsida, Lycophyte; then Sphenopsida, Sphenophyte; then Pteropsida, Pteridophyte. So Xylopsida is the primitive plants. First, vesicular pteridophytic plants. In these plants, homosporous condition may be protostele is present. Example, xylot. Next one, Lycopsida. So best example, Lycopodium. Lycopodium and Selaginella. Lycopodium and Selaginella, both homosporous and heterosporous condition. In Lycopodium, homosporous condition. In Selaginella, heterosporous condition. Then Equisetum. Examples for Spinopsida, Equisetum. Except Equisetum, in Spinopsida, all are fossil plants. Next one, Pteropsida, Bryopteris. Then Pteris, Adiantum. Examples for Pteropsida, Filipophyta, Bryopteris, Pteris and Adiantum. These all are homosporous condition. Advanced pteridophytic plants, more evolved pteridophytic, highly distributed pteridophytic plants are this Pteropsida plants, Bryopteris, Pteris and Adiantum. So Adiantum generally is known as a walking fern. Adiantum is known as a walking fern. Why? Because when tips of the adiantum leaves are touched to soil, they give birth to new individual plants, play important role in vegetative propagation, vegetative reproduction. That's why this is known as a walking fern. So adiantum is known as a walking fern. Why? Because when tips of the leaves are touched to soil, then produce a formation of the new individual plants. That's why this is known as a walking fern. Some characteristic features of fern plants, that means Pteropsida plants. So generally in these plants, root system is a adventitious root system. Adventitious roots are present. Then stem is present in underground of the soil like a dark rhizome. Dark rhizome. Rhizome is present in Adiantum, Bryopteris and Pteris. So it will useful for vegetative reproduction. Next one, leaves. So generally simple leaves are pinnately compound leaves are present. Pinnately compound leaves. The petiole of the leaves, young leaves, stems is covered by brown color hairs. These hairs is known as a ramenta. Brown hairs is known as a ramenta. Ramentas are present in Bryopteris, pteridophytic plants. Brown color hairs. Next one, young leaves shows the circinate vernation. Circinate vernation. Young leaves shows the circinate vernation. That means young leaves are spirally coiled condition is known as a circinate vernation. Next one, male gamete sperms are multiciliated, multiflagellated condition, spirally coiled condition. These are some characteristic features of fern plants. Next one, economic importance of pteridophytic plants. So pteridophytic plants generally ornamental plants. So these plants are useful as a ornamental plants. Example, Adiantum, Pteris, Bryopteris, etc. So Adiantum, Pteris and Bryopteris are useful as a ornamental plants. Then these plants are due to binds the soil. It prevents the soil erosion. These are prevents the soil erosion. So pteridophytic plants plants will bind the soil. Due to binds of soil, these are prevents the soil erosion. Then pteridophytic plants are useful as a medicinal purpose. So pteridophytic plants useful as a medicinal purpose. Best example, adiantum. So adiantum has an anti-helminthic property. Anti-helminthic property. Adiantum has the anti-helminthic property. Then some pteridophytic plants are useful as a food source. For example, Marsilia. Some tribal peoples are using this Marsilia as a food source by extraction of a starch. Next one, Equisetum. Equisetum plants will useful for scouring and polishing metals. Equisetum will useful for scouring of utensils and polishing the metals. Then Azula will useful for nitrogen fixation. Azula will useful for nitrogen fixation. Useful as a excellent biofertilizer. Excellent biofertilizer. Azula is a smallest pteridophyte. Azula is a smallest pteridophyte. Smallest pteridophyte is a Azula act as a excellent biofertilizer. So these are the economic importance of pteridophytic plants. Then life cycle of pteridophytic plants. Example, Dryopteris. So pteridophytic plant body is a sporophytic condition, diploid in a condition. Then this forms the sporophyll. In sporophyll, a group of sporangia is known as a sori. A group of sporangia is known as a sori. Each sori may contain 2 to 6 sporangia. In some pteridophytic plants, all sporangia grouping to form a compact of structure. This compact of structure is known as a strobili. This compact of structure is known as a strobili or a cone. Selaginella and Equisetum. Selaginella and Equisetum. In Selaginella and Equisetum, cone or strobili is present. Generally, these pteridophytic plants are growing in moist, shady localities. But Selaginella can grow in xerophytic habitat also. That means xeric condition. Some pteridophytic plants grow in xeric condition. Best example, Selaginella. So by the grouping of all sporangia form a compact unit of structure. This is called strobili or a cone in Selaginella and Equisetum. Then sori. These sporangia contain spore mother cells. Spora mother cell, diploid condition. Then spore mother cells undergoes to meiotic division. Meiotic division. Results of meiotic division from the spores. Haploid spores, that means microspores and mega. Then by the germination of spores, here form the prothallus, haploid gametophyte. On prothallus may be male sex organ, female sex organ, antheridium and archegonium is present. Antheridium, then archegonium. So haploid condition. Then antheridium will produce the antherozoids. means sperms. Archaeopterygium produce the egg cell. Then by the fusion of this egg cell and sperm, syngamy zygote, then zygote to embryo to sporophytic plant body. Sporophytic end condition, then gametophytic end condition. Sporophytic and gametophytic shows the heteromorphic alternations of generation, heterologous alternations of generation. So life cycle of Bryopteris.